Hello friends, it's Bhaktin next episode. So in this episode, we'll be continuing with the NCERT quick reference. And chapter is Molecular Base of Inheritance for 2. And remember, daily I'm putting 2 questions in ACE Botany at 4:15 p.m. as a practice question for NEET exam. Detailed note I already published in YouTube for most of the chapters in Botany from NCERT. All references based on March 2024 Reprinted edition NCERT textbook. For convenience, I will share the screen so it will be easy for you. We can also save time since some statements I already wrote there. I think screen came. So we can proceed. We can start with the introduction. Main topics, we will be discussing introduction, transforming principle, biochemical characterization of transforming principle, genetic material is DNA, find established. Now introduction, search for genetic material. See, even though Frederick Mayer's work was there, Mendel's work was there, which is the original genetic material still was not clear. Some experiments step by step later confirm which is the genetic material. You will remember the term nuclein connected with the scientist Frederick Mayster in 1869. And the term nuclein was converted to nucleic acid by Altman. Now we can continue with the transforming principle. I think up to that it is clear. So we can proceed here. Transforming principle. Okay, transforming principle. Scientist Frederick Griffith, 1928, he was a British bacteriologist. So everything I wrote written there for convenience. And his experiment giving a clue or a guessing that DNA may be the genetic material. Now, experimental part of the area, that is starting with the organism. Streptococcus pneumoniae, commonly called pneumococcus. 2 strains, S3 and R2. S means smooth, and that is capsulated or shiny, pathogenic or virulent. R means rough, non-capsulated, non-pathogenic, non-virulent. And the second organism taken was rat or mice. I think up to that it is clear. Now, S3, R2, then their different combinations were injected into mice, then they analyzed it. I think first page is clear, so we can proceed to second page. So this is the list connected with NCERT. S strain injected into mice. That is, this is causing mice will die. R strain injected into mice. R is rough, no pathogenic. So mice will live. Here it is dying, here it is living. S strain heat kill injected into mice, again mice live. Now the next combination, S strain heat kill. So it is having no life, but DNA is there. Live R strain. So this combination. And when you injected into mice, mice dies. And that is due to change in the genetic material. That phenomenon is called transformation. So conclusion for this experiment is heat kill S strain bacteria converts live R strain bacteria into live S strain. This conversion may be due to transfer genetic material from heat killed S strain to live R strain. Still, the biochemical nature of genetic material was not able to define from this experiment. So next step now came biochemical characterization of transforming principle that is done by 3 scientists, Oswald Avery, Colin MacLeod, McCarthy, 1933, 1944. See, initially the scientists have different views regarding genetic material. Some scientists even told that it may be protein up to that level. Later, Avery, MacLeod, McCarthy worked on biochemical aspects of transforming principle connected with the Griffith experiment, its biochemical aspects. And they proved that the genetic material is DNA. But not all biologists were not at all convinced. But all biologists. So some biologists convinced, but some other biologists not convinced. So not all biologists were not convinced. I think idea clear. Some believed it, some not accepted it. So we need a proof. So first we can check the experimental part of this experiment, biochemical characterization. So what they done? They purified biochemicals like proteins, DNA, RNA from the heat-killed S cells to see which one could transform live R cell to S cells. And they discovered that DNA alone from S bacteria caused R bacteria to become transformed. And this is given in NCERT. Now the diagram, that is general view I given there to get an easy understanding. Experiment to prove that DNA is the transforming material, Avery, MacLeod, and McCarthy. In A, what they done, culture medium plus RNA of S type. So no change. Second one, culture medium plus proteins of S type. No change. Third one, culture medium plus DNA of S type. But remember, enzyme DNA is added. So no change because DNA will be digested. Now fourth one, culture medium plus DNA of the S type. That is, this is causing some R bacteria transformed into S type. That is how the they proved it S time. And what is meant by DNA? That is what I given here. DNA means enzymes that can digest DNA. ASE means suffix representing enzyme. Biomolecular maybe studied it. Plural of DNA is DNAS. So difference is these two. I think I made it clear. As usual, after this experiment, many scientists not accepted. Thus, a confirmation test become necessary. That is the next experiment. That is how final proof came. It's an important one. Evidence from experiments with the bacteriophage. That is otherwise, this experiment is otherwise called unequivocal proof. Otherwise called blender experiment. Blender means mixing. Done by Arthur Hershey and Martha Chase in 1952. Aim is to show that DNA is the genetic material. Now, organisms, virus, T2 bacteriophage, infects E. coli. T2 phage, viruses have two components, one is protein and one is genetic material DNA. Here, two chemicals, that is, we can easily check DNA or protein is the genetic material. Now, phage DNA labeled with the phosphorus 32, protein code with the S35. So P32 DNA, protein code S35, and radioactive phosphorus and sulfur. And this is a famous NCERT diagram you can find, bacteriophage. Radioactive S35 here, P32 here. And three steps were there. One is infection. What is meant by infection? Three steps. Infection, here both types of labeled phages were allowed to infect bacteria with the S35 and P32. Now blending. Blending means these bacterial cells were agitated in a blender, that's an instrument, blender, to break the contact between virus and bacteria. And third step is centrifugation. Thus, the virus particles were separated from bacteria by spinning them in a centrifuge. These are three steps, important one. And here, that is with the S35. After this experiment, they detected no radioactivity in the cell, but they are able to detect in supernatant because this DNA, this S35 or DNA, means that protein S35 came out. So radioactive S35 detected in supernatant after centrifugation. While with the P32, here it is P32, that gets incorporated with the DNA. So after blending, centrifugation, etc., we can find radioactive P32 detected in cells and no radioactivity detected in supernatant. That means that DNA or P32 ended into DNA. Here S35 ended into membrane-like structure or supernatant. I think I did clear. So clear experiment. And this is the unequivocal proof for DNA is the genetic material. I think these three experiments clear: transformation, biochemical aspects, then final unequivocal proof. Now we can proceed to the next page. So thus, it was clearly established that the DNA is the genetic material. Now, what are the features of genetic material? Then DNA versus RNA, RNA world, etc. These all aspects we'll be discussing in the next episode. I think this experiment is very important for theory entrance. Most simplest manner given it for quick reference. So within 5 to 6 minutes, you can finish it. That is the advantage of quick reference. Detail not already, it may take more time. So this is for quick reference. This way, small segments I am publishing. So it will be easy for you. If you like this episode, subscribe, base button, like it, share it, comment on it. We can meet later. So take care. Bye. Thank you.